So far we have understood that MLK overexpression will cause necroptosis of the pancreatic tumor cells, and that is beneficial. However, in this video we will discuss how the experimental results that shows that the necroptosis that are driven by MLKL will actually also promotes metastasis in the liver, metastasis of pancreatic cells in the liver, and which is accompanied by macrophages. So let us go through the results that are presented in this figure. So in the panel 1, first they have checked the invasion properties or invasion capabilities of the tumor at different stage, that is at T1 M0 stage and T1 M1 stage. So what they have found out that at the T1 M0 stage the invasion characteristics are lower, and the tumor volume is seems to be higher, whereas at T1 M1 stage the tumor aggregates are smaller but the invasion characteristics seems to be higher, so indicating that at T1 M1 stage the tumors has more invasion properties compared to the T1 M0 stage, or the small aggregates has more invasion properties. And these things they have validated from the fluorescence images, not fluorescence images, from the macroscopic images, the PANC1 and ASPC1 cells at different condition. First, when the MLKL are overexpressed, then they can see that number of cells are reduced because of the necroptosis, both in the case of PANC1 and in the case of this ASPC1. However, when these inhibitors of MLKL are used, that is GW, they do not see a decrease in the cells. However, they see the increase in the number of cells compared to the control. And when they use both the overexpressed MLKL and the inhibitors, they can see the number of cells again goes down in the case of PANC1 and also in the case of ASPC1 compared to the control, indicating that the MLKL uh, indicating that this MLKL overexpression will cause this necroptosis. And when they need to check the proliferation and the growth characteristics, they use this PCNA, which is the marker, which is the indications, which PCNA, which is the antigen for the proliferation capabilities. And when they use the MLKL, and what they have found out when they use this, when they don't use the inhibitors, they can see the reduced signals for PCNA, indicating that there are less proliferation characteristics in the PANC1 cells. But when we use both this MLKL overexpression and this uh, MLKL overexpression and the inhibitors for the MLKL, they can see that increase in the proliferation characteristics. And similar results they have observed also for this ASPC1, that is when both overexpression of MLKL occurs and the inhibitors of MLKLs are there, they see the more proliferation characteristics. And this is the panel D, which where they examines the tumor growth rate, and this is how they conduct the experiment. That is, each well in the 96 well plate will have 2000 tumor cells, and they allow 24 hours for tumor spheroid formation, and then measure it using the high content cell quantitative image analysis system. And what they have find out, when the overexpression of MLKL are there, they see the reduced tumor volume, but at the same time they see the low proliferations rate. And in the case of where MLKL are overexpressed and inhibitors are used, they see that the tumor volumes are uh, in line, or there is not much difference between the tumor volumes that has MLKL inhibitors and tumor volume that has MLKL overexpression and MLKL inhibitors both. That can be seen here both in the case of PANC1 and ASPC1. But this in the last conditions has more proliferation characteristics. So this smaller tumor volumes has more proliferation characteristics. The quantification of these fluorescence images are in the right hand side of the panel D, where we can see that there is a statistical difference between this MLKL overexpression and the normal vector. However, there is no statistical difference between MLKL overexpression plus inhibitors and the MLKL overexpression plus inhibitors, and there is only inhibitors. So this is evident both for this PANCO1 and ASPC1. Thereafter, after validating that the smaller tumor volumes or smaller this tumor spheroid has more proliferation characteristics, they decided to do their studies on the two different mice models. One that has a weak immune system, that is NOD, SCID mice models, and one that has high immune system, that is C57BL6. In the case of uh, in the case of this mouse model where it has low immune system, they they see the decrease in the tumor volume. They see the decrease in the tumor volume, that is indicated by this red dot, the decrease in the tumor volume when the MLKL are overexpressed. However, when they check it for this C57BL6, that is the mice that has high immune system or efficient immune system, they see the increase in the tumor volume when the MLKL are overexpressed, indicating that MLKL's overexpression might be interacting with the immune system in a way that promotes the metastasis of the tumor. And these things are quantified in the right hand side of the panel E, where we can see that when the mice are uh, in the C57, uh, they see that when the MLKL are overexpressed, when the MLKL are this is the liver metastasis models in the tumor lesions, that is this is the spin signals. They injected the tumors in the spleen of the mice. So this fluorescence intensity is decreased when they use MLKL overexpressions only. However, for the spleen they do not see. For I think the spleen they are talking about this side. When it comes to the spleen, they see the decrease in the fluorescence intensity. That indicates that the tumors are spreading or not present at the place of the injection or at the place of where the tumor is injected, that is at spleen. However, when they check the liver, what they have find out that in the case of this C57BL6, that is mouse that have high immune system, efficient immune system, will have more fluorescence intensity, indicating that the tumors volume are metastasis into the liver in the case of high immune system. And when they check these mice models, what they have find out is that for the spleen they have find out that when there is no MLKL gene, there are no less macrophages, whereas when there are more MLKL gene, the immune system in terms of the macrophages interacts with the MLKL gene, that is indicated by the this triangle. And it shows that macrophages are in contact with the high expression of MLKL at the spin, that might promote metastasis somehow. And when they did their experiment in vitro, uh, not in vitro, when they did their experiment in vivo with these macrophages inhibitors, they can see that reduction in the fluorescence intensity that can be seen here. The fluorescence intensity are reduced compared to the case where there is no inhibitors, no MLKL inhibitors. And same things they have observed both for the spleen and for the liver. For the liver also, when there is no inhibitors for the macrophages, they see that increase in the tumor volume at the metastasis. But when there is a when there is inhibitors for macrophages and also for the MLKL, not for the MLKL, only for the macrophages. When there is inhibitors for macrophages and overexpression of MLKL, the metastasis got reduced. And these things that they have validated using the in vitro experiment where they co-culture PANC1 with macrophages and ASPC1 with macrophages. So when only the MLKL are overexpressed and there is no inhibitors for macrophages, then they see that the metastasis or large spheroid formations. But when they use the inhibitors for MLKL or MLKL overexpressions with its inhibitors, they see that decrease in the spheroid formations is comparable to the vector. And same things they have observed for the ASPC also. That is, when the MLKL overexpressed, they see that more large spheroid formation or metastasis are more. However, when they use the MLKL inhibitors, the metastasis reduces and it's comparable to the original vector. And when they use the orthopedic model in this immune system, they find out that tumor volumes are spread across the in the liver when the MLKL are overexpressed. That can be seen in the panel I. And when they did the experiment on multiple mice models, what they have find out that when the MLKL are overexpressed, there are almost all the cases in the they did the experiment with 5 cases. In all the 5 cases, they find out that it will cause liver metastasis. And when there is no overexpression of MLKL, they see that only 20% of the cases, or in the 1 out of 5 mice models, have liver metastasis. And in the they check the tumor volume, they find out that the tumor volumes are low when the MLKL are overexpressed, and this is in line with their panel D results. That is, smaller tumor volume has more metastasis. The tumor weights are also in line with the tumor volume. That is, MLKL overexpressions has lower tumor weight compared to the where there is no MLKL overexpressed. And when they check the liver metastasis at different conditions, that is, when the macrophages are also inhibited along with the MLKL overexpression, they find out that in the 25% of the cases they see the liver metastasis, whereas when the MLKL are inhibited, they see that only 20.5% has the 12.5% of the cases shows liver metastasis. But when the MLKL are overexpressed and there is no inhibition for macrophages, around 87.5% shows metastasis to the liver model, to the liver. In the case of this C57B L6 orthopedic mice model, where these pancreatic cancers are injected using orthopedic. Pancreatic cancer cells are injected in this C57B16 mice models, or the orthopedic models, where the xenograft of the pancreatic cancer cells are injected into this mice model. So I hope you understood how this one of the positive characteristics of MLKL driven necroptosis helps in the metastasis in liver of the pancreatic cancer cells, which are accompanied by the macrophages. If you need to understand their experimental results again in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.